Hi there! After transplantation, patients need immunosuppression, otherwise they will reject their organ. In this lecture, we will learn about the different agents, the indications, the targets and the possible side effects. So to understand the targets of immunosuppression, we first have to understand the alloimmune response. This movie helps you and shows you step by step the main pathways involved. So sometimes you might think it's a little complicated, but you do not have to know all details. Try to think in pathways. That will help when we focus on the drugs. So let's now look at the movie.
Initiation of the alloimmune response takes place when alloantigen-specific T cells come into contact with professional antigen-presenting cells expressing or presenting the alloantigen recognized by the T cell. This contact mainly occurs in the secondary lymphoid organs of the recipient. It is this cognate interaction which triggers T cell activation. The T cell receptor specifically recognizes the alloantigen in the form or in the context of MHC class I or class II expressed on the antigen-presenting cell, thereby receiving signal 1. However, for full activation, the T cell should also receive signal 2 or co-stimulation. CD80 and CD86 on the antigen-presenting cells cross-link the CD28 receptor on T cells. Together, signal 1 and 2 result in efficient signaling through the T cell receptor CD3 complex, inducing several signaling cascades, including a rise in intercellular calcium. The interaction of calcium with calmodulin will activate the phosphatase calcineurin. Upon activation, calcineurin will dephosphorylate the transcription factor, nuclear factor of activated T cells, or NFAT. Upon dephosphorylation, NFAT can translocate from the cytoplasm to the nucleus and bind to the promoter region of the interleukin-2 gene, amongst others, resulting in an enhanced transcription of this cytokine. Increased expression of interleukin-2 mRNA will result in translation of the interleukin-2 gene into protein and secretion of interleukin-2 by the T cells. Secreted extracellular interleukin-2 or IL-2 binds to the IL-2 receptor and induces a signaling cascade of various kinases, including the PI3 kinase pathway and the molecular target of rapamycin, or mTOR pathway, which initiates the cell cycle. Lymphocytes require synthesis of purine and pyrimidine nucleotides for replication as building blocks for DNA. T cell activation results in proliferation, which leads to a large number of T cells with identical specificity.
So you've now seen the signaling mechanisms of an alloimmune response, which is of importance to understand the targets for treatment. Immunosuppressive agents are used at the different stages. In the first place, as induction therapy in the first week to provide strong suppression of the immune response. Secondly, as maintenance therapy, which is used continuously to prevent acute rejection. And third, as rescue therapy in the case of an acute rejection episode. So take a minute to look at examples which are used as induction, maintenance and rescue therapy. Let's now look at the targets of the most important agents used. You might think, why do we have to know this? Well, the drugs are the reason why we have made such improvements in transplantation. Knowing the cellular targets can help in understanding the basis. In addition, it might give us insight into side effects. Like I mentioned earlier, It's not necessary to know all details, so try to think in pathways.
Tacrolimus binds to a family of intracellular receptors termed the FK506 binding proteins to create a new complex that blocks calcineurin. Calcineurin dephosphorylation of NFAT is inhibited, preventing NFAT to enter the nucleus. A CTLA-4 Ig fusion protein called belatacept is a potent antagonist of the B7 ligands. It binds with high affinity to the CD80 and CD86 present on antigen-presenting cells, thereby preventing the interaction of these molecules with CD28 on the T cells. This so-called co-stimulation blockade results in efficient inhibition of T cell activation. Basiliximab is a chimeric anti-interleukin-2 receptor monoclonal antibody. It specifically binds to and blocks the alpha chain of interleukin-2 receptors, also known as CD25 antigen, on the surface of activated lymphocytes. As a consequence, interleukin-2 cannot induce proliferation of these cells. Sirolimus and everolimus engage FKBP12 to create complexes that bind to and inhibit mTOR. Inhibition of the target of rapamycin blocks signal 3 provided by cytokine receptors, thereby preventing the induction of cell cycle progression. Mycophenolic acid, the active compound of CellCept, inhibits inosine monophosphate dehydrogenase. This is the rate-limiting enzyme in de novo synthesis of guanoside nucleotides required for DNA synthesis. T and B lymphocytes are more dependent on this pathway than other cell types. Azathioprine is converted into 6-mercaptopurin in the body, where it blocks purine metabolism and DNA synthesis. It most strongly affects proliferating cells, such as activated T and B cells. Depleting antibodies, including alemtuzumab, antithymocyte globulin, and rituximab, bind to and deplete lymphocyte populations. Alemtuzumab is a humanized monoclonal antibody that selectively binds CD52, which is expressed on a large array of lymphocytes, including T and B lymphocytes. Antithymocyte globulin, or ATG, is a polyclonal antibody preparation directed at many antigens present on thymocytes and lymphocytes. For both antibody types, binding to these cells results in depletion through various mechanisms, including complement-dependent cytotoxicity, antibody-dependent cell-mediated cellular cytotoxicity, or ADCC, and apoptosis induction. Depletion only occurs after a certain threshold of antibody binding is reached. Rituximab is a chimeric monoclonal antibody recognizing CD20, an antigen specifically expressed by B cells. The selective depletion of B cells occurs through similar mechanisms of complement-mediated lysis and ADCC.
Well, you've just seen that there are many different targets of the drugs, all with the same aim, to suppress the immune system. What about the results? And what about the side effects? Current regimens of immune suppression yield excellent patient and graft survival at 1 year. However, the long-term survival rates are not optimal yet, so the agents display major side effects, including an increased risk for infections and malignancies. And some agents may be nephrotoxic itself and may promote cardiovascular disease. In Module 4, we will focus on these long-term effects. It's always a challenge to find the right balance between under and over immune suppression. So what have we learned in this lecture? Immune suppression is necessary in the transplant recipient. They can be divided in induction, maintenance and rescue therapy. They have different modes of action. Interference with the alloimmune response plays a central role. And lastly, current immunosuppressive agents have major side effects. In the next lecture we will discuss cellular therapy as a possible novel treatment in transplantation.